I am extremely honored to introduce our host for today, Dr. Leonard Sachs. Dr. Sachs is the founder of CITC and has been the mastermind and driving force behind it. He joined FDA in 1998 as a medical reviewer in the Office of New Drugs. Subsequent positions included acting director of the Office of Critical Path Programs and associate director for clinical methodology in the Office of Medical Policy in the Center for Drug Evaluation and Research. In this capacity, he has led efforts to support novel approaches to clinical trials, including the use of electronic technology. Besides his involvement in the design and analysis of clinical trials, he maintains a special interest in tuberculosis and other tropical diseases and has published and presented extensively on these topics. Please help me to welcome Dr. Leonard Sachs.
Thank you very much, Brenda, for the kind introduction. Hello to everybody. I'm Leonard Sachs from the Office of Medical Policy in the Center for Drug Evaluation and Research at FDA, and I'm delighted to welcome you to this year's Clinical Investigator Training course. As you all know, clinical trials are the bedrock for the approval of medical products, and clinical investigators are amongst our most important partners in developing the evidence used for regulatory decisions. We rely on you for all the clinical data that we receive to support the approval of new medical products, and we are tremendously appreciative of the critical contribution that you make to medical science and to the health of our society. This clinical investigator training course is an important opportunity for us at FDA to reach out to you with the goal of giving you a broad understanding of the science and the regulation of clinical trials. As many of you know, we have been running this course for years, and we are very encouraged by the tremendous support we have received. Today, as Brenda said, we have more than 8,000 registrants participating from all over the world, representing a global community dedicated to the development of the highest quality medical products. Our 3-day program covers a broad range of topics, including clinical trials, their design and analysis, the determination of drug safety, ethical considerations, and some of the innovative approaches to make trials more efficient and accessible to participants. We've included lectures on non-clinical drug development, animal studies, toxicology, chemistry, manufacturing controls, and we have an extensive session to address regulatory requirements, trial quality, and investigator responsibilities. We've divided the course into approximately 4-hour sessions spread over 3 days, and the idea is to cover the most topical areas of interest to investigators. Today, we will focus on selected topics related to the design of clinical trials. Tomorrow, we will look at some of the non-clinical and early clinical data that you may come across in the investigator's brochure. And on Thursday, we will discuss the responsibilities of investigators and investigator site inspections. By sharing our experiences inside FDA, we're hoping to provide you with an insight into FDA requirements that will give you as investigators greater confidence and expertise in conducting clinical trials, and that will improve the clinical trial enterprise in general. I wanted to take this opportunity to thank our speakers in advance, all our colleagues of mine at FDA who have generously given their time to participate and prepare these presentations. I apologize for not being the most technologically gifted myself, but I hope we will be able to give you a satisfying experience as free of glitches as possible. During the panel sessions, we hope you will send in your questions to make this as interactive as we can. So without further ado, I'd like to introduce the speakers in our first session. Kim Smith will begin with an overview of FDA, its structure and mandate. Jim Smith will cover the basics of clinical trial design, and Mark Levinson will review the statistical principles for drug development. So Kim, over to you.
Thank you for that introduction. For the next 20 minutes, I'm going to provide you with an overview of the FDA Structure and Mandate to provide some background for later topics that we'll cover during the course. So the objectives of my talk are to provide a brief history of the FDA, to describe the current mission of FDA, and to introduce FDA's legal and regulatory framework for drugs and biologics. So to start with a brief history of the FDA, I think it's important to recognize that the agency was built on a legacy of public health failures, so unsafe, ineffective, counterfeit, or adulterated drugs, as well as some unethical practices and fraud. The image on the right is an advertisement for Mrs. Winslow's soothing syrup, which was marketed in North America and the United Kingdom in the 19th and early 20th centuries as a cure-all medicine for fussy babies. But the primary ingredients were morphine and alcohol, and thousands of children are believed to have died from overdoses or from morphine addiction and withdrawal. The lower right is an image from the New York Times. This is on the Tuskegee syphilis study, which was started in 1932 by the U.S. Public Health Service working with the Tuskegee Institute. It was a study to record the natural history of syphilis. Participants were not consented and were told that they were being treated for bad blood. By 1943, penicillin was the treatment of choice for syphilis and was widely available, but the participants were never told about this or offered treatment. In 1972, the study was exposed when an article was published by the Associated Press, but unfortunately at that time, men in the study had been untreated for many years and their disease had progressed. To continue with our brief history, I'll provide an overview of just some key milestones in U.S. food and drug law. So starting with 1848, we had the passage of the Drug Importation Act, which required U.S. Customs Services to inspect entry ports for the entry of adulterated drugs from overseas. It's really one of the earliest attempts to regulate the U.S. drug supply. Moving forward to the early 1900s, at that time, diphtheria patients were routinely treated with an antitoxin that was derived from the serum of horses. The horse serum was manufactured locally and there were no central or uniform controls. In 1901 in St. Louis, Missouri, the blood of a tetanus-infected horse was used to develop the antitoxin and was administered to 13 children who ended up dying of tetanus. This tragedy led to the 1902 passage of the Biologics Control Act, which allowed for the first controls over processes used to produce biological products, including vaccines, serum, and antitoxins. In 1906, we had passage of the Pure Food and Drug Act, which prohibited interstate commerce in misbranded and adulterated foods, drinks, and drugs. In 1912, we had passage of the Shirley Amendment. The Shirley Amendment prohibited labeling medicines with false therapeutic claims intended to defraud the purchaser, but it was a standard that was very difficult to prove. Moving forward to the late 1930s, in 1937, we had another tragedy. At the time, sulfanilamide was a drug that was used to treat streptococcal infections, and there was demand to provide it in a liquid form. So a company experimented and found that sulfanilamide dissolved in diethylene glycol, compounded the product and sent it all over the country. Unfortunately, diethylene glycol is antifreeze and is toxic. The product killed 107 people, many of whom were children, and really provided a dramatic example of the need to establish safety before marketing a product. This tragedy led to the passage of the 1938 Food, Drug, and Cosmetic Act. The act required that new drugs be shown to be safe before marketing and really started a whole new system of drug regulation, and it was the origins of much of what we do today. In 1944, we had the Public Health Service Act. The Public Health Service Act covered a broad spectrum of health concerns and included provisions related to the regulation of biological products. In 1951, we had the Durham-Humphrey Amendments. Durham-Humphrey Amendments defined the kinds of drugs that could not be used safely without medical supervision and restricted their sale to prescriptions by licensed practitioners. So this was our first foray into prescription products. We then have our next tragedy. Thalidomide in the 1950s and early 1960s was widely used to treat nausea in pregnant women. It became apparent in the 1960s that thalidomide was resulting in severe birth defects in thousands of children, limb deficiencies that you can see in the images on the screen. This was primarily occurring in Western Europe, and fortunately, the product had never been approved in the United States. Dr. Francis Kelsey, who was a FDA medical officer, had concerns about the amount of safety data that was available and had kept the drug off of the U.S. market. This example provided a lot of public support for stronger drug regulation and led to the 1962 passage of the Kefauver-Harris drug amendments. These amendments ensured drug efficacy and also enhanced safety controls. For the first time, drug manufacturers were required to prove to FDA that their product was effective before the product was marketed. In 1968, we had the Drug Efficacy Study Implementation, or DESI. Basically, many products had been approved between 1938 and 1962 on the basis of safety alone, and the agency had to generate a process to go back and review 4,000 plus drugs to determine whether they should stay on the market. The products that did stay on the market went through this DESI approval process. So fast forwarding a bit to the FDA now, the agency has over 18,000 employees, many of whom are scientists. Recent numbers are that over 11,000 of these 18,000 employees are in science fields. The agency oversees the safety of more than $3.6 trillion worth of food, tobacco, and medical products, accounting for 21 cents of every dollar spent in the US. On the right, you see a figure that was from 2023, although remains largely current. It shows that the human drugs program is the largest by far, followed by foods, tobacco, devices and radiological health, and then biologics. This course will really focus in on the human drugs and biologics programs. So I'm going to shift focus a bit to provide some information about the legal framework that guides the work that we do at FDA. Our work is grounded in statute with the Federal Food, Drug, and Cosmetic Act and Public Health Service Act providing our statutory authority for oversight of clinical investigations to evaluate safety and effectiveness. The laws are further explained through regulations that the agency issues. They can be found in the Code of Federal Regulations. This is how we implement the statutory authority over the conduct of clinical investigations. A few key sections. Title 21 is Food and Drugs, and Chapter 1 addresses the Food and Drug Administration. Subchapter A provides some general provisions. This is where you'll find information, for example, on informed consent and institutional review boards. Subchapter D addresses drugs for human use, and Subchapter F addresses biologics. FDA guidance is also issued regularly by the agency, and FDA guidance is intended to be advisory. It's intended to assist regulated entities in complying with the regulations and to help them to understand FDA's current thinking on a topic. There are numerous guidances, many of which are relevant to investigators, others might be more relevant to sponsors, and if you're interested in whether there's guidance on a particular topic, the link at the bottom provides the search location. So much of the agency's work revolves around review of submissions to certain types of applications. The first is the Investigational New Drug Application, or IND, and then you have the New Drug Applications, NDA, or Biologics License Application, or BLA, and I'll talk a little bit more about each of these. So current federal law requires that a drug be the subject of an approved marketing application before it's distributed across state lines. Because a sponsor probably will want to ship the drug to clinical investigators in many states, it needs to seek an exemption from that legal requirement. And an IND is the means through which they technically obtain that exemption from the FDA, which allows them to ship the product across state lines. This, of course, allows them to initiate clinical studies in humans. There are three main types: investigator-initiated INDs, which I think would be the most common in most situations where you're studying a drug to meet an approval standard. There are also emergency use INDs as well as treatment INDs. There are two categories. There are commercial INDs. These are the ones that are initiated by a sponsor who intends to eventually seek some sort of a labeling change or approval of a product related to the studies that are conducted under that IND. For studies that are being conducted more for academic or research purposes, there are also research INDs. All INDs must include preclinical data, manufacturing information, as well as clinical protocols and information on investigators that will allow the agency to evaluate whether the study that's proposed is reasonably safe to proceed. Not all studies of drugs need to be conducted under IND. There are exemptions for specific types of studies. So first, the product has to be lawfully marketed in the United States already. This cannot be a new product that is not already on the market. The study cannot be intended to support a new indication or a significant labeling change for the drug. It cannot the study itself cannot involve a route of administration, a dose, a patient population, or some other factor that would significantly increase the risk to participants in the study. And the investigation needs to be conducted in compliance with other regulations for instance, institutional review board, informed consent, and promotion. So what is the purpose of an IND? The IND provides the data needed for FDA to ensure the safety and rights of clinical study participants are maintained, and also to ensure the quality and adequacy of studies intended to provide evidence of the safety and effectiveness of medical products. After a sponsor submits an IND opening study protocol, the agency has 30 days to review the protocol, at which point the protocol will go into effect unless the sponsor is notified earlier by FDA that the study may proceed or FDA issues a clinical hold. When the agency reviews these INDs or reviews any submission to an IND at any point in a development program, one lever we have if we have concerns about the study is to issue a clinical hold. This is an order that's issued by the FDA to delay a proposed clinical investigation or to suspend an investigation that is already ongoing. When we issue a clinical hold, no new subjects can be recruited or given the drug, and the patients in the study need to be taken off therapy unless they're permitted to continue by FDA in the interest of public patient safety. For instance, if stopping the drug abruptly would be harmful, there can be a plan to manage that in particular situations. Not all clinical holds are complete. There are partial clinical holds. For instance, if you have enough data to support lower doses but not higher doses, there may be a clinical hold on administering higher doses. Or for instance, if you have enough data to support studies in adults but not children, the studies could be allowed to proceed in adults while there's a clinical hold on studies in children. So what are the grounds for a clinical hold? For phase 1, 2, and 3 studies, the agency could find that there is really unreasonable and significant risk of illness or injury to participants in the study. There may not be enough information to understand the risk, so there may just be more information needed before a particular study or part of a study could move forward. The investigator brochure, which is intended to provide information to investigators on what is currently known about the drug, may be misleading, erroneous, or incomplete and need to be corrected to make sure that information on the drug is being accurately and completely portrayed to investigators who are enrolling patients in the study. The clinical investigators may not be qualified to conduct the investigation. And then lastly, if there is a disease that is life-threatening and affects both genders, but one gender, often women, are being excluded, that could be another ground for a clinical hold. For phase 2 and 3 studies, all of the above apply, but in this case, the agency can also put a study on clinical hold if the protocol is believed to be deficient in design to meet the objectives of the study. So moving on, once you have completed all of your studies under IND, you're ready for a marketing application. As I mentioned, there are two main types of marketing applications. One is a new drug application under the Food, Drug, and Cosmetic Act, and the other is a biologics license application under the Public Health Service Act. Both are a mechanism to provide the FDA with information that's needed to support the marketing approval of a new product, or to provide additional information to support a new indication or changes to labeling for an already approved product. Just to take a bit of a step back to a couple of basic definitions now that we're getting into new drug applications and biologics license applications. A drug is defined in the Food, Drug, and Cosmetic Act as articles recognized in the official United States Pharmacopoeia, official homeopathic pharmacopoeia of the United States, or the official national formulary. And they're articles that are intended to use in the diagnosis, cure, mitigation, treatment, or prevention of disease in man or other animals. And they're articles other than food intended to affect the structure or any function of the body of man or other animals. And then finally, they're articles intended for use as a component of something specified in A, B, or C. A biological product is a little bit different. It is defined in the Public Health Service Act as a virus, serum, toxin, antitoxin, vaccine, blood component, etc. Again, that's applicable to the prevention, treatment, or cure of a disease or condition in human beings. So all biological products would also meet the definition of a drug. So what do we receive in a marketing application, meaning an NDA or a BLA? First is chemistry, manufacturing, and controls information. So how is the product manufactured? How will it be packaged, et cetera? Non-clinical pharmacology and toxicology information, what is known about toxicities that were seen in preclinical studies or animal studies, human pharmacokinetics and bioavailability studies. So what is known about the clinical pharmacology characteristics of the product? Of note, later in this course, you'll have lectures on all three of these topics from some of the experts in these areas. For anti-infectives, there's information on the microbiology of the product. And then, of course, there's clinical information from studies in humans, clinical trials, and other types of studies, statistical information, as well as information related to potential pediatric use of the product. We get samples, as well as information on product labeling. And then for participants in trials, we do get case report forms describing events that may have happened during the studies, as well as data sets where we can actually reproduce some of the analyses necessary to confirm the efficacy and safety findings of the study, as well as to conduct additional analyses if needed. And then finally, we receive legal information. So information from the applicant about patents, exclusivity, financial certifications, and so on. Product. So the key decisions before us when we get either an NDA or a BLA are whether the drug is safe and effective in its proposed uses, and whether the benefit of the drugs outweigh the risks of the drug, whether the labeling is appropriate and what it should contain, so providers who read the label understand how to use the drug and who it would be beneficial in. And then finally, whether the manufacturing methods used to maintain the drug's quality are adequate and that the methods preserve the drug's identity, strength, quality, and purity. And so at the end of the day, if we find all of these to be true, we will approve a drug and it will be legally marketed at that point. I'll move on to the challenge question. So the first challenge question is for a drug that is lawfully marketed in the United States, an investigational new drug application is never required to conduct a clinical investigation using the drug. Is this true or false? So this is false. Not only does the drug have to be lawfully marketed in the US, but to be exempt from IND requirements, the study can't be intended to support a new indication or significant labeling change. It can't involve a route of administration, dose, patient population, et cetera, that would significantly increase risk. And the investigation needs to be conducted in compliance with other regulations, such as IRB and informed consent requirements. So challenge question number 2, which of the following is not a reason for a clinical hold? A, the investigator brochure is incomplete. B, there's insufficient information to assess risk. Or C, the investigation of a drug for disease that manifests in childhood excludes children. Or D, clinical investigators are not qualified, which is not a reason for a clinical hold. So C is not a reason for a clinical hold. Just to briefly summarize my talk, FDA has been working to promote and protect public health for over 100 years. It's one of our oldest consumer protection agencies. The laws, regulations, and guidance provide a framework for the development and approval of drug and biologics and really frames the work that we do day to day at FDA. INDs are the mechanism through which FDA evaluates whether clinical investigations are reasonably safe to proceed. And NDAs and BLAs are the mechanism for our oversight of the US sale and marketing of drugs and biologics. Thank you very much for your attention. I will be back a bit later during the question and answer period, but now I will turn it over to Dr. Jim Smith.
Thank you very much, Dr. Smith. So as Kim said, my name is Jim Smith. I am the Director of the Office of New Drug Policy and the Office of New Drugs at FDA. And during this session, I'll be speaking for the next 45 minutes about the basics of clinical trial design. The learning objectives are to describe the characteristics of adequate and well-controlled studies, to describe the purpose of control groups and various types of controls, and to describe methods to reduce bias in clinical investigations. So to begin, and this might seem really basic, but we should keep in mind that the purpose of doing clinical research is to ask important questions and to answer them with appropriate studies. So the quote on this slide is the first sentence of the regulation that describes adequate and well-controlled studies, which are the characteristics of studies that should be submitted to FDA to demonstrate effectiveness for a drug. And it leads right off with a purpose. The purpose of conducting clinical investigations of a drug is to distinguish the effect of a drug from other influences, such as spontaneous change in the course of the disease, placebo effect, or biased observation. And although this regulation was specifically written to address, investigations seeking to demonstrate effectiveness of a drug, like I mentioned, the same principle applies to any study, frankly. One is generally interested in discerning the effect of some independent variable itself, like here the drug, on one or more dependent variables. That is the outcomes of interest. And if something other than the intervention being studied could have plausibly caused the outcome, then the study will have failed to answer the scientific question. So here I'm going to summarize at a high level the characteristics of an adequate and well-controlled study, which, as I just said, are good scientific principles to consider regardless of the type of clinical research that you're involved in. And I'm going to go into several of these points in the coming slides, but let me run through the list as an overview. First, studies should have clear objectives. So these should reflect the research questions, they should be clear and explicitly stated in a protocol, along with a summary of the methods that will be used to analyze the data to answer the questions. The study should use a design that permits a valid comparison with a control group, and that provides a quantitative assessment of the effect of the intervention. So I'll talk about examples of control groups shortly, as well as some common designs that are used to compare two or more groups in various ways. The selection of trial participants should be done in a manner that provides adequate assurance that they either have the disease or condition that's being studied, or in the case of a drug or intervention intended to prevent or reduce the risk for some outcome, the participants should at least be susceptible to having that outcome. Otherwise, there'd be really no reason for them to be subjected to the drug being studied. The method of assigning patients to the treatment and control group should minimize bias. This is where randomization usually comes into play for studies with a concurrent control, which I'll discuss in a bit. And beyond the method of treatment assignment, adequate measures should also be taken to minimize biases on subjects, observations, and analysts. Blinding is an example of such a measure. Next, there should be well-defined and reliable assessments of how the individuals in a trial may respond to an intervention. That is, what outcome or outcomes are being measured? Are we certain that we're measuring what we intend to be measuring? And last, there should be analyses of the results of the study that are adequate to assess the effects of the drug. These should generally be pre-specified in the protocol or statistical analysis plan before the study begins. So this may seem obvious, but it really can't be overstated. The scientific questions to be answered and the trial objectives need to align. So without having a really clear, specific question or set of questions, the study design choices are likely to be suboptimal, and then its results might be insufficient to answer the questions that you're trying to answer. So the questions that are driving the research should really inform choices about trial design, data collection, and analysis. So for example, if a new drug is being compared to a drug already on the market, is your question whether the new drug is clearly more effective than that existing therapy? Or would you also be interested in knowing whether it's at least not materially worse than the existing therapy, even if it's not clearly better? So each of those questions would not only require distinct analysis plans, but it would also impact sample size and possibly the importance of blinding and endpoint selection and other considerations. Or as another example, is the objective to understand the effect of treatment when it's added to a specific background treatment, or would it be sufficient to understand its effect when it's added on to standard of care, which may or may not include a certain background treatment? The choice of a control group is always a critical decision. Control groups allow discrimination of patient outcomes caused by the test treatment from outcomes caused by other factors. So if the course of a disease were always the same in a given patient population, or if it was completely predictable from patient characteristics, then the observed outcomes that occur in a single treated group of patients, that is like a single arm trial, could simply be compared with the outcomes that you know would have occurred in the absence of that treatment. And that is possible in certain circumstances, but typically a concurrent control group is needed because it's not possible to predict outcomes with adequate accuracy or certainty. And the choice of which control to use is important because it will affect the inferences that can be drawn from the trial, potentially the ethical acceptability of the trial, the degree to which bias can be minimized, the type of participants that can be recruited and how quickly, the types of endpoints that could be studied, the credibility of the results, and so on. So here are the 5 types of controls that are generally recognized in adequate and well-controlled investigations and are mentioned in the regulation that I cited. The first 4 are usually concurrent controls. That is, the control group and the test group are chosen from the same population and treated as part of the same trial. So the first, which I'm sure you've heard of, is the placebo control. So in a placebo-controlled trial, participants are randomly assigned to the test treatment or to an identical appearing treatment that does not contain the test drug. And it's very important to recognize, and sometimes this seems to be misunderstood, that the use of a placebo does not imply that the control group is untreated. In many placebo-controlled trials, the new drug and the placebo are each added to a common background therapy or other standard of care regimen. These trials are almost always double-blind, meaning that the patients and the investigators don't know what the treatment assignment is. And combined with the use of randomization and blinding, which I'll discuss a little bit more in a bit, this design actually controls for many, if not all, potential influences on the outcome other than those arising from the drug itself. So, for example, many diseases wax or wane in severity. So if a participant improves significantly after randomization, was that destined to happen regardless of the intervention, or, just because of the natural course of the disease, or was that a drug effect? Having a placebo arm can give you information about that. Or you might have heard about regression to the mean, where patients are eligible for a trial because they have, let's say, high blood pressure. So a high blood pressure reading gets them into the trial, but that might have been kind of a random high for them just from day-to-day variability. And in those cases, over time, even if without treatment, their blood pressure would usually fall back to that patient's average. And so having a placebo group can help assess for that and can show, what would have happened to a group in the absence of treatment. The second example is the no treatment control, and this is exactly what it sounds like. Participants would be randomly assigned to test treatment or nothing at all. That is not even a placebo. So really the main use of this design is when it's difficult or impossible to blind the treatment assignment. In the third example, participants are generally randomly assigned to different doses of the same drug or different regimens of the same drug. If a higher dose of the drug is shown to be more effective than a lower dose, that can provide really strong evidence that that higher dose is effective. The limitation, of course, is that the result would tell you something about that high dose, but if you haven't also included a placebo arm, you won't know whether or not the low dose is effective. Or similarly, if you don't see a difference between the high dose and low dose, then you're left wondering, are they both effective or are they both ineffective? For these reasons, it can be beneficial to include a placebo arm, which then provides information about a dose of 0, if you will. Next, in an active control trial, participants are generally randomly assigned to either the test treatment or a known effective treatment, which is referred to as the active control or the positive control. These trials can be double-blind by having participants in each arm receive not only their assigned treatment, but also a placebo version of the other arm's treatment, and that's referred to as a double dummy technique. So blinding in these trials is not always possible, though, because of some combination of either the number of interventions or different regimens or different routes of administration and so on. Last, in an externally controlled trial, a group of participants who received a test treatment are compared with a group that is external to the study. So maybe it was a group of patients observed at some time in the past, which is then referred to as a historical control, or it's a group observed around the same time, but in a different setting entirely, like a disease registry or a natural history study, or information collected from medical practice, and so on. Now, on occasion, an effect observed in a single-arm trial is known without question to be something that would not have occurred spontaneously. And so although this is sometimes called a baseline controlled study because a value on treatment is compared with the individual participant's baseline value, that's actually also a type of external control, because what you're really doing there is you're relying on the very strong knowledge, which is derived external to the trial itself, that the change that's been observed would not have occurred in the absence of the intervention. And there is a lot of interest right now in externally controlled trials, but it's really beyond the scope of my presentation today to cover them in sufficient detail. Suffice it to say that the lack of an internal concurrent control arm and randomization and blinding require exceptionally careful consideration of various ways that bias could be introduced into the trial and whether it's even possible to sufficiently mitigate these limitations to result in an interpretable trial. Okay, so moving on to the study population, that is, which patients are going to be included in the trial. So typically, the eligibility criteria or the inclusion and exclusion criteria should identify patients with or at risk for the disease or condition being studied. And this population might be more narrowly defined or more broadly defined depending on the objectives, which could vary during drug development. So early stage drug development, perhaps the population is being more narrowly defined because you want a more homogeneous population to detect a signal of a drug effect, or perhaps there's too much uncertainty about the safety of a drug in the early stages of drug development that you need more exclusion criteria. But as that drug progresses to later stages of drug development, it becomes increasingly important to define the study population more broadly to more closely represent the diverse populations for which the drug is intended. And in general, you should avoid unnecessary exclusion criteria. So both to facilitate enrollment as well as generalizability of results, like each exclusion criterion should be very well considered and believed essential. And you should think about even commonly used exclusion criteria if they're actually necessary. Why exclude patients with renal impairment if the pharmacokinetics of the drug have been determined for those patients? Maybe they could be included, but with a drug with a dose adjustment, for example, or an age-related exclusion should be very well justified. So overall, the population that's expected to benefit from answering the research questions of interest ought to be represented in the clinical investigation, and enrollment goals should be proactively set to achieve that. So related to the study population, I'd like to briefly touch on the concept of enrichment. So this is the selection of a study population in which detection of drug effect, if one is present, is more likely than it would be in an unselected population. So this is a design feature that narrows the study population in certain ways in hopes of improving the ability to detect the effect of a drug. And so I'll go through three broad categories of enrichment strategies. So first are strategies to decrease variability. because reducing variability often makes it easier to detect a signal among the reduced noise, if you will. So one could aim to reduce between patient variability or within patient variability. And an example of the latter is what I've included here. So a placebo lead-in period could be used in which all patients are provided a placebo for some amount of time before randomization. And those who show a large response to placebo, or those who demonstrate that they're very poorly adherent to taking the assigned placebo, could be excluded before they're ever randomized, as those participants might be even, might be more likely to introduce variability during the trial, making it more difficult to identify a drug effect. Another enrichment strategy is prognostic enrichment. So the question here is, who's most likely to progress or have an outcome of interest? So let's say you're conducting a trial in which the outcome of interest is hospitalization for heart failure, and the trial is designed to run until a certain number of such hospitalizations have occurred. Well, it could be quite beneficial to enroll patients who are at greater risk for hospitalization, like maybe those who have had a hospitalization for heart failure within 1 year of enrollment, or some other characteristic that informs their prognosis, which is why it's prognostic enrichment. The last strategy I'll mention is predictive enrichment. So here, the question is, who's more likely to respond to the drug than others without the factor that's being used for selection? So prognostic enrichment considers who's most likely to have an outcome regardless of treatment. And predictive enrichment considers who is predicted to respond to the drug. And so a randomized withdrawal study is an example of this. So here, all the patients are placed on the drug for some period of time, and then a certain subset of those who believe they've responded to the drug by some predetermined criteria are then selected to be randomized to continue on the drug or to switch to a placebo. And that's an enrichment strategy because all of the patients who don't think that they're responders to the drug will have been weeded out, if you will. And they won't then be subjected to the test versus control randomized portion of the trial. Another example of predictive enrichment, maybe it's even more straightforward, is let's say you're enrolling patients who have a tumor tissue that's positive for a certain biomarker, such as a certain gene variant, and the drug's mechanism of action specifically targets that gene variant. That would be another example of predictive enrichment. Enrichment strategies can limit the generalizability of the findings, and I think Dr. Levinson might touch on this in his talk that's coming up. Okay, next we have method of assignment to treatment arms or study arms. This should be done in a manner that minimizes bias and ensures comparability of the groups with respect to pertinent variables. So typically, in a concurrently controlled study, assignment is by randomization. So why is randomization important? Let's think about a couple scenarios here. So in a randomized study, a patient who meets the eligibility criteria gets assigned to a group by a completely random method. So I'm illustrating that here by a flip of a coin, if you will. So heads, they get drug A, tails, they get drug B. So coins aren't actually used, but I think you get the point. So now think about an observational study where you go to the medical records of patients who are getting drug A and patients who are getting drug B, and you record things that you find in the record about their medical course, and then you compare the outcomes of interest of those who got drug A versus those who got drug B. So, well, why could that be a problem? Well, just highlighting one concern for illustration of this point, consider how these patients each came to be on drug A versus drug B. So they may have come to their healthcare provider who took a whole host of things into consideration when making a treatment decision. Maybe the patient's age, their comorbidities, concomitant medications, family history, labs, the patient's own preferences, their insurance status, and so on. And if a difference observed between outcomes for drug A compared with drug B, if a difference is observed between those two, it's plausible that some other factor or factors other than the drug itself is associated with the selection of which drug to prescribe, but also with the outcome itself. So this could lead to an incorrect conclusion that there's a difference between drug A and drug B, when in fact, the difference is the result of the underlying patient populations. And this is referred to confounding, and I think that Dr. Levinson might be discussing this as well. So assuring that the subject populations are similar in the test and control groups is best attained by randomly dividing a single sample population into the test and control groups. So randomization is going to avoid systematic differences between groups with respect to both known and unknown baseline variables that could affect the outcome. And the balance of unknown baseline variables is an important one to keep in mind. It's not uncommon to see a great deal of hand-wringing over various baseline characteristics and their apparent balance or lack thereof in a randomized trial. And sometimes that is justified, especially when there's this known strong prognostic or predictive characteristic. But often it's not. In part, it should be kept in mind that there are a very, very large number of unmeasured baseline characteristics, and we have zero idea about their balance. Or lack thereof, because we have no idea what they, or their values are. Of course, this doesn't mean don't trust a randomized trial. In contrast, appreciate the ability of randomization to avoid these systematic differences between groups. And perhaps even more important, recognize that randomization provides a sound basis for statistical inference. And the reasons for that are beyond the scope of this presentation, but it really is a very important outcome or a reason for randomizing participants in a trial. So it's important that the control group be as similar as possible to the test group, except for the intervention that's being studied. To best assess the effect of the drug itself, the similarity between groups should be both at baseline as well as after baseline, through the study. And as I just discussed, randomization facilitates the similarity between groups at baseline. But many differences can arise after baseline, differences in adherence to assigned treatment, the use of rescue therapy perhaps, completeness of data and follow-up, and differences in how the outcome assessments are conducted, reported, and so on. And in the next few slides, I'll talk about methods to reduce bias entering the study during the trial itself. One of the key methods to reduce bias, at least in a randomized study, is to conceal the treatment assignments, which is referred to as blinding. So blinding is intended to minimize the impact of potential biases that could be conscious or unconscious. And as I just said, the aim here is to ensure that the study groups are treated and observed similarly during the trial, except for the intervention of interest. So what impacts could the knowledge of treatment assignment have? Well, I'll give several examples on this slide, but it's certainly not all inclusive. And I'm also not saying that these will arise in every open label trial, but these are the types of things that should be considered and mitigated if possible. So healthcare providers could treat the trial participant differently if they know what study arm they've been assigned to. And those differences in how they're treated could directly affect the outcome of interest. So this would be another example of confounding. Those on active drug might report more favorable outcomes, such as responses on a questionnaire because they expect they should improve on the new drug being tested. Observers may be less likely to identify and report treatment responses in the control group, like the no treatment group. Those on active drug may be more or less likely to report adverse events. And this is a good reminder that oftentimes you don't know which direction these biases may go, right? It's unpredictable. Knowledge may affect rigor of attempts to obtain follow-up data. And knowledge could impact treatment decisions related to analysis. Now, this isn't the investigator or the patients, but this could be a way that knowledge of treatment assignment could affect those other personnel, like those who are analyzing trial data. So that could impact their decisions, such as looking for reasons to justify excluding a poorly performing site, if getting rid of the results might make the rest of the trial results appear much more favorable for the test drug. And as I mentioned, there's no simple test for any of these. You can't really look after the fact and say, oh, well, this bias must have crept into the trial. And sometimes it'll favor the test drug, sometimes not. And you can't really assume that that's going to all balance out. So taking measures to reduce bias, especially in circumstances when blinding isn't possible, is really important. Now, even with blinding, it's important to realize that patients or investigators might be able to detect or guess their treatment assignment. Maybe there's a distinct adverse effect, that's only observed with the test drug, for example, or something similar to that. And in such cases, especially if the study is open label, one should consider pre-specified decision rules, blinded outcome assessments, and other measures. So as an example of a blinded outcome assessment, if the endpoint of an open label trial requires the histological assessment of tissue, or maybe the rating of a skin lesion from a photograph, the individual conducting the assessment should not be made aware of the treatment assignment. And this doesn't prevent all bias from entering the study, of course, but it would be an attempt to mitigate a biased assessment. Again, whether conscious or unconscious, by the study personnel who are aware of what treatment the participant is receiving. Knowledge of interim results, whether it's at the level of the individual or the treatment group, also has the potential to introduce bias or otherwise influence the conduct of the study and interpretation of its results. So special attention should be given to maintaining the confidentiality of interim results. And finalizing statistical analysis plans before treatment assignments are revealed is another mechanism to ensure that knowledge of the data itself does not influence the choice of analyses, or the choice of the inclusion or exclusion of certain data, the weight that's placed on certain endpoints, and so on. So shifting gears a bit, next I'd like to talk about assessing response in a clinical investigation, which is often described as the endpoints of the study. So the choice of endpoints should be aligned with the study objectives. And if the study is being conducted to answer the question of whether a drug is effective for a certain disease or condition, those endpoints should be meaningful for the intended population, and may also take into account the views of patients. The primary endpoint should provide relevant evidence related to the primary objective of the study. So, and typically the sample size of a trial is calculated based on information related to this endpoint. And then secondary endpoints are usually either supportive measurements related to the primary objective. That is, they provide additional information that might give one greater confidence in the results observed on the primary endpoint, or they may be measurements related to secondary objectives of the study. Usually the primary endpoint and at least some secondary endpoints are included in an analysis plan that protects against making false conclusions that an intervention has an effect when in truth it does not. Exploratory endpoints are those that may further explore or support study findings, or they may explore and generate new hypotheses for future research. So exploratory endpoints would generally not be included in the analysis plan in a manner that protects against making false conclusions. And because many potential endpoints and analyses could be explored, there's the very real chance that a difference between groups could be observed on one or more of these endpoints solely by chance alone. But what is an endpoint exactly? So this is a definition that comes from the current version of the FDA NIH BEST resource. BEST stands for Biomarkers, Endpoints, and Other Tools. So an endpoint is a precisely defined variable intended to reflect an outcome of interest that is statistically analyzed to address a particular research question. So when thinking about an endpoint, obviously you need to identify the assessment itself. For example, is it a lab value? a patient-reported outcome like a questionnaire, a physiologic test like maybe a pulmonary function test, or a clinical event like a stroke. Next, the endpoint might incorporate other information that occurs during the trial. So for example, if the endpoint is defined in terms of a success or failure, perhaps participants who discontinue their assigned study treatment prematurely are automatically considered a treatment failure. It should consider the timing of the assessment. So for example, are you interested in a hemoglobin value after 6 months of treatment? Or maybe the assessment is one that could happen at any time, such as a participant having an event like a stroke, and really the time of interest is the duration from randomization until when that first stroke occurs. And finally, if relevant, consider how assessments within each participant are formulated or combined. So for example, sometimes a composite endpoint is used, which means that multiple distinct events can be combined, if you will, into a single endpoint. Or maybe several assessments are made during the window of time, like a daily diary over a 2-week period. And you might decide in advance that you will take the average of all 14 scores, or maybe it's the maximum or the minimum, to represent the endpoint assessment in a single value. So there are different types of endpoints, and I've already alluded to several, but a couple major categories include clinical outcomes and surrogate endpoints. A clinical outcome is an outcome that describes or reflects how an individual feels, functions, or survives. That is, it directly represents the clinical state of an individual. A surrogate endpoint, on the other hand, is an endpoint that is used as a substitute for how a patient feels, functions, or survives. So an effect on the surrogate endpoint is expected to predict an effect on the ultimate clinical outcome of interest. And the level of evidence that an effect on the surrogate predicts such a clinical benefit can vary, of course. So if the evidence is strong, at least in drug development, that's called a validated surrogate endpoint. Examples of this could include reductions in hemoglobin A1C, which have been clearly shown to predict benefit with respect to microvascular complications and diabetes, or reductions in blood pressure, which reduce the risk of certain adverse cardiovascular events, such as stroke, among patients with hypertension. I'd also like to mention the idea of intercurrent events. So intercurrent events are events that occur after treatment initiation that affect either the interpretation or the existence of the measurements associated with the clinical question of interest. So some examples of these types of events could be treatment discontinuation, and that can occur for a variety of reasons, like adverse events, perceived lack of efficacy, other things. Beginning a new therapy or switching to other treatments, and which could affect the outcomes of interest, or even something like death. And it's important to note that intercurrent events do not lead to a problem of missing data. So missing data is data that would be meaningful for the analysis, but were not collected, which is different than data that do not exist or data that are not considered meaningful because of an intercurrent event. So for example, although missing data strategies is beyond the scope of what I'm talking about in this session, imputation techniques are often used to impute values for those that should have been collected but weren't. So consider a participant who dies during the trial before they reach the time point at which the endpoint would be measured. Well, they won't have an endpoint value, but it would usually not make any sense at all to impute a value for someone who died, essentially pretending that they were alive and had an endpoint measured. So in this case, death is an intercurrent event. And if it's predicted to happen with some frequency in a certain study population in a trial, how to handle such an intercurrent event should be considered and incorporated into the design and analysis plan before the trial even begins. So you should also consider possible intercurrent events when defining the clinical question of interest. So here's an example. In a trial of a drug for type 2 diabetes, Suppose the primary endpoint is the change in hemoglobin A1C from baseline to week 24. And suppose the trial allows for rescue medication if certain thresholds are met with respect to blood glucose values being too far out of control, for example. So here are two examples of possible treatment effects that may be of interest. What's the effect of the new drug if rescue medication is not available? Or what is the effect of the new drug regardless of whether rescue medication is used? Now, I don't have time to discuss how the effect of interest would result in different design considerations, but to suffice it to say that these are distinct questions, right? And that should be considered on the front end, not after the fact. Okay, so the elements that I've discussed can be put together in various ways, and I'll just touch on a few here. The most common study design for a randomized controlled trial is the parallel group design. So in this design, participants are randomly assigned to different groups that are then followed in parallel, if you will, until the endpoint. Another study design, which still employs randomization, is the crossover design. So here, participants are randomly assigned to an initial study treatment, so test versus control in my illustration here. And after some period of time, endpoints are assessed. And then there's usually a washout period where no treatment is given, allowing the effects of the first period to, possibly return to their baseline values. And then within each of the initially randomly assigned groups, each participant then receives the other study treatment for a second period, and the endpoints are assessed again. And this design can be more efficient than the parallel group study and therefore require fewer patients because it has the advantage of each patient serving as their own control, which removes the between patient variability from the comparison between treatments. But there are several things to consider that might make this a suboptimal design. So first, there's the possibility of a carryover effect, which is the effect of the treatment from the previous time period carrying over and influencing the response during the subsequent time period. And that washout period in the middle is an attempt to mitigate this, but it's still possible that the previously administered treatment may have altered the individual in some way so that they'll react differently to the treatment from that time onward, or maybe the washout period is just, an insufficient amount of time or whatnot. Or maybe the outcome is some sort of a functional test that requires some skill, so that participants are typically perform better during period 2 on the task because they were trained in a sense during period 1. So those are things to watch out for. Second, the design is generally used when the observation periods are relatively short, so that the endpoint needs to be one that responds and can be measured fairly quickly. Also, since ideally each individual would return to their baseline condition before each period, it's generally used in chronic diseases where the outcome is not dependent on the time of the assessment. So last, I'll describe a 2x2 factorial design. So I've drawn the study arms here as a table, but this would typically be a parallel group study. But each participant gets randomly assigned, so in this case, to either placebo, drug A alone, drug B alone, or the combination of drug A and drug B. And of course, this could be blinded using the double dummy technique that I mentioned earlier. And this allows you to essentially study 2 interventions using the infrastructure of one trial. So first, you can assess the effect of drug A by comparing the 2 groups in the left column, that is placebo and drug B, with the 2 groups that include drug A, that is drug A alone and drug A plus drug B. Similarly, you can assess the effect of drug B by comparing the top row, so placebo, placebo and drug A, so it does not include drug B, to the bottom row, which does include drug B. But the design also allows other analyses. So for example, you can look at whether drug A and drug B each contribute to effectiveness when they are used in combination. You can also check whether the effect of drug A is modified in the presence of drug B and vice versa. Factorial trials can certainly include more than 2 interventions, but it can quickly become infeasible since the number of cells in a full factorial trial will simply become too large. Also related to the comparison between groups, and I've mentioned this already, is the question of whether you're testing a superiority or a non-inferiority hypothesis. So in a trial of drug A versus a known active control drug B, the superiority hypothesis is testing whether drug A is better than drug B. And by better, this would usually mean more effective, although I suppose you could test a hypothesis related to superior safety as well. In contrast, the non-inferiority hypothesis would be testing whether drug A is not meaningfully worse than drug B. And the non-inferiority margin, or NI margin, quantifies what is meant by meaningfully worse. Now, if no difference is detected between drug A and drug B in a trial, then there's a question. Does that mean that both drugs are effective or that both drugs are ineffective? And that's obviously a very important difference. And so because there's no confirmation, within that trial itself that the active control drug B is effective, that's an assumption. And so it must be considered very carefully. So assay sensitivity is the ability of the trial to have detected a difference between treatments of a specified size. So said another way, assay sensitivity means that, had the study included a placebo, the active drug would have been shown to have the effect that was assumed. And this is not directly assessed within a non-inferiority trial. So you need to consider such things as, historically, have consistent findings been observed with the active control, especially when compared against placebo in those past trials? If there has been a consistent effect, then you have greater confidence in the active control in the new trial. Second, how similar is the new trial to those historical trials where the active control drug was tested against placebo? Is the patient population similar? Were there important differences in concomitant treatments that have become available? Are the differences in how the study endpoints are defined or analyzed? Is the active control being dosed similarly in the new trial as it was in the historical trial? So these are things to consider because you're making an assumption that the active control's effect is constant between the new trial and the historical trials. And that's referred to as the constancy assumption. Now, third, the quality of the new trial needs to be excellent. So poor conduct can reduce the ability to detect differences between groups because sloppy conduct generally introduces variability. So this can be problematic in a non-inferiority trial because the trial could be considered a success if the new drug and active control appear to behave similarly. And you won't know for sure whether that means that both drugs are effective or both drugs are ineffective if the trial was not conducted well. Finally, just a couple high-level comments about trial design and to some extent conduct. So efforts should really focus on activities that are essential to the study. So for example, it's far more important to expend energy and resources to ensure that the primary endpoint is assessed exactly as intended, that follow-up is as close as possible to 100%, rather than pouring resources into ensuring that every scrap of source data is available for the 50th exploratory endpoint. Obviously, I'm giving an extreme example there, but I'm just trying to emphasize that you should consider eliminating non-essential activities in data collection to increase quality by simplifying conduct, improving the efficiency of the study, and to really target resources to the critical areas. So deploy your resources to identify and prevent or control errors that matter. And last, clinical study design is best informed by input from a broad range of interested parties. So patients, patient organizations, study coordinators, site staff, clinical investigators, like these, people who will be directly involved in the successful completion of the study are very well positioned to inform its design. patients may take one look at a protocol and say that it's far too burdensome in a variety of ways, and that could potentially foreshadow a lot of premature treatment discontinuation if it's not modified. Clinical investigators and site staff could give a lot of insight into the feasibility of enrolling patients who meet the inclusion and exclusion criteria, and maybe those need to be modified or broadened. Or whether the scheduled study visits and procedures are overly burdensome and might also lead to early dropouts. So, and patients and patient organizations can inform the general relevance of study endpoints, study settings, and some among other things. So seek this information out and really consider using it to optimize the design of the study. So with that, I'd like to move on to my challenge questions. The first is, which of the following is not an example of enrichment? Enroll patients with a lab value that indicates they are at greater risk for the outcome of interest during the trial. Limit enrollment to patients with a certain genetic variant that is specifically targeted by the investigational drug. Ask trial participants who demonstrate poor adherence after randomization to withdraw from the trial. And require patients to have 3 consistent values during the screening period for the assessment of primary interest in the trial. Give you a few seconds to think about the answer. And C is not an example of enrichment. So A is an example of prognostic enrichment, B, predictive enrichment, D is a way to reduce variability, but C says to ask participants who demonstrate poor adherence after randomization to withdraw from the trial, and you do not want to do that because after randomization, pulling participants out with poor adherence could introduce bias. And so if you're going to use this technique, it should be done prior to randomization. The second challenge question, the choice of a control group affects which of the following? A, the inferences that can be drawn from the trial. B, the ethical acceptability of the trial. C, the degree to which bias in conducting and analyzing the trial can be minimized. D, the scientific credibility and impact of the results. And E, all of the above. And hopefully that's clear that that should be E, all of the above. So to summarize this session, you should design clinical investigations to distinguish the effects of an intervention from other influences. Always start with the clinical questions of interest, and the design of the clinical study should flow from those. Key design aspects include the study population, an appropriate control, well-defined endpoints, measures to minimize bias, including the assignment of treatment, and the analytical plan. And with that, I'd like to thank you very much for your attention, and I will turn it over to Dr. Levinson.
Good afternoon, everyone. My name is Mark Levinson. Today I'm going to be talking about statistical principles for clinical development. There are 2 learning objectives to my presentation. The first is to understand basic statistical concepts such as bias, variability, and multiplicity. And the second is to understand the role of statistical concepts in good study design, conduct, and analysis. So basically, the first learning objective will lead into the second. Here's my outline. First, I'm going to give a quick review of good study principles. Dr. Smith has already presented on much of this. And then the heart of my talk is I'm going to go into statistical concepts and the implications on study elements. And this will start with a discussion of the distinction between bias and variability and the implications of each. And then I'm going to drill down to variability and then bias. And finally, I'll provide some concluding statistical principles. Okay, just to get some terminology down that I'll be using throughout my presentations, I'd like to provide some terms for the stages of a study. The stages start at the design stage. This is the conception, planning, and specification of the study. Typically, this will result in the protocol and statistical analysis plan, or SAP. The second stage is the conduct. This is the running of the study. This includes recruitment, screening, intervention, concomitant care, outcome ascertainment, and safety and study monitoring. The next stage is the analysis of the study. This is the number crunching, going from the raw data to the final results. Typically, this is where people believe the statistician comes into play, but I argue that the statistician has a role in all the stages of a study. Finally, there's reporting. The reporting of the study should basically align with the protocol and statistical analysis plan. I won't go into reporting much today, but there are guidelines on this. A common theme or message in my presentation is that design and conduct are more important than analysis. In other words, analysis cannot make up for poor design and conduct. One should focus on good design and conduct, and the analysis will be straightforward. And statistical concepts motivate good design and conduct, and that's really the point of my talk, is to show where statistical concepts can improve design and conduct and result in overall a better study. So what are the goals of study design, conduct, and analysis? The first goal is very clear. You're trying to address a specific clinical question. That's why you're running the study. I'm going to present two additional goals that come into play as you choose design and conduct decisions. The first one is to minimize bias and minimize variability. I'm going to spend a lot of time on what that means, both for bias and variability. And finally, you have to be able to ethically, safely, and feasibly conduct a study. You can't do a study that's not ethical for patients, not safe for patients, or it's just not feasible. Now, the problem is there's some conflict among these goals. There's almost inevitably some conflict among these goals. And let me give you an example. Okay, here's an example of balancing these three goals. And this is the concept of enrichment versus generalizability. So the clinical question we're interested in is a drug effect on cardiovascular events for all patients with type 2 diabetes. And the emphasis here is on all patients. This is an event trial, so we're trying to measure cardiovascular events. Event trials usually require a certain number of events to get the necessary statistical precision. You can get more events for the same number of patients if you study patients with additional CV risk factors. For example, patients that may have had a prior heart attack. So for the same number of patients, if your patients are at greater risk, you're going to be able to run a smaller trial. However, you're not really answering the first clinical question that I stated because you're interested in all patients with type 2 diabetes. So here we see that there's a balance between feasibility and answering the clinical question. Okay, that was my quick review of good study principles. I'll return to many of these principles throughout the talk. And now I'm going to get into the heart of the presentation, and that is the statistical concepts and their implications on study elements. And first I'm going to define bias and variability and the important distinction between the two. Okay, here we have four bullseyes. Let's start in the upper left, low variability and low bias. The dots are intended to represent your data points, and the center of the bullseye is intended to represent the truth, the answer you're trying to seek. So in this bullseye on the upper left, we see how data points are packed closely together. That's the low variability, and they're actually centered on the center of the bullseye, and that's the low bias. Moving to the upper right, we see that the data points are spread apart, and that's the high variability, but they're generally centered on the average around the center of the bullseye, and that's the low bias. Moving to the lower left, the data points here are clumped together with low variability, but they're far from the center of the bullseye. So this represents high bias. And finally, the combination of the two in the lower right, we have the data points spread apart and not really centered around the middle of the bullseye, and that's high variability and high bias. So those basically informally define these two terms. Now the problem is you don't get to see the bullseyes. So say these were your data points, and I asked you the question, do these data have low bias? Well, I mean, let's say, are we able to even answer, do they have low variability? Well, that's a relative term, but using kind of the scale we had on the last slide, let's say these data points have low variability. But because we don't have the bullseye here, we don't know the true answer, because that's why we're running the clinical study. We don't know if these data points are centered on the truth or centered around some value away from the truth. So we don't know about the bias in this case. I hope those few slides demonstrated a few things. First of all, I hope they demonstrated what bias and variability ultimately are, but there are some other important concepts I hope they demonstrated. First, that we see and can measure variability. We can see how the data points are spread apart from each other. We can measure that using such measures as standard deviation and ranges. We do not see bias. We don't know what the true answer is, or not we wouldn't be running the study. Bias is mainly addressed in the design and conduct stages, and I hope the statistical principles I bring out through this talk will help motivate good design and conduct. Now we have a definition of bias and variability, and I'm going to be drilling down into each of them. First, variability and associated statistics and concepts related to that. So variability and sample size. So sample size, of course, is the number of people in the study. I'm going to ask you a question. What is a better estimate of the average age of this session's attendees? Now, there may be over 1,000 attendees in this class, and we would like the average of those 1,000 values, but we're going to take a sample of the attendees, as is often done. So the first possibility is we pick a random sample of 5 attendees and calculate their average age. The second choice is to pick a random sample of 50 attendees and calculate their average age. Now, which of this is going to give you a better average age estimate of the overall session attendees? Clearly B will, because more people are involved in this sample. Some people will have high values relative to the overall mean, some will have low. And the more you pick in your sample, the more there's going to be cancellation, and there's going to be less variability in these larger sample sizes. So now I'm going to introduce a little notation to further explore variability in sample size. Let's call x hat 5 the mean of 5 people, and x hat 50 the mean of 50 people. We intuitively saw on the last slide that x hat 50 has less variability than x hat 5. Let's look at variability in sample size a little more mathematically. The standard error of x hat 50 is a measure of the variation of x hat 50. And the standard error is defined as the standard deviation of x. Those are the individual values of x, the individual ages in the population, divided by the square root of n, the sample size. So as you can see from this formula, as the sample size n gets larger, the denominator gets larger and the ratio gets smaller. So for larger sample sizes, the standard error decreases. Okay, we're going to get into a little bit more math. I'm going to introduce the z-statistic. The z-statistic is the effect estimate. So that's basically what you're trying to estimate, like the effect of a drug, over the standard error of the effect estimate. So this is an estimate over a standard error. It's basically a signal to noise quantity. Just to make this a little concrete, I'm going to assume the effect estimate is 0.5 meters. Now I'm going to be using kind of a running example through a few slides that the endpoint we're interested in is how far you can walk in 6 minutes. And what we've observed in this study is that there's a 0.5 meter greater distance that people on drug can walk than on control. That's a small amount, but that won't be important right now. As the sample size increases, we saw on the last slide that the standard error goes down. And as the standard error goes down, because it's the denominator in this quantity, the z statistic goes up. And what I won't really show is as the z statistic goes up, the p-value goes down. What that means is it's more significant, the same result, the 0.5 meters, if you observe that in a larger study, that will give you a more significant result. Let's continue this. Typically, if there is no drug effect, that the probability of seeing the absolute value of the z statistic greater than 2 is 0.05. So 0.05 is 1 in 20. And absolute value means if we see -3 or +3, they're both greater than 2. If we observe an absolute value of the z statistic greater than 2, then we say the finding was significant at the two-sided 0.05 level. So what this says is if there was no drug effect and we observed, say, a z-value of 3, that was a pretty unlikely event, certainly less than 1 in 20. And that gives us doubt that there really is no drug effect, that maybe the assumption of no drug effect is wrong. So we call that statistically significant. It's a tricky concept, but that's basically what it is. Now let's return to the effect of sample size. For a given real effect, say 0.5 meters, the probability of a significant effect increases with sample size. So we saw that on the last slide. If the effect is 0.5 from a larger study with more sample size, it will have a smaller p-value, more significant than the same effect from a small study. So when we say a larger study has more power than a smaller study because the probability of seeing a statistical significant result for the same effect size is larger. So to say that again, larger sample sizes have higher power. Now I will move on to the concept of multiplicity, also known as multiple bites from the apple. Suppose I told you a study found a drug had a statistically significant effect for females aged 51 to 60 years old. Well, that sounds like an important finding. Now suppose I told you the study actually studied people from all ages, from the first year of life to people over 90, and both males and females. And they calculated the z statistic for every combination. So each of these cells gives you a chance of the z statistic being greater than 2, even if there's no drug effect. So perhaps it's not surprising that some were greater than 2. In fact, greater than 2, even if there's no drug effect, occurs 1 in 20 in a 1 in 20 chance. So here it's not at all surprising that if we looked at enough of these different combinations, one of them would be greater than 2. But it doesn't, I think, call into question that there's no drug effect. It very likely could be no drug effect, and you would see a result like this. So multiplicity, even if a drug has no effect, if you conduct enough statistical tests without proper adjustment, you'll find some results statistically significant. This is known as multiplicity. And what the implication is here, that although they're nominally statistically significant, they're not really statistically significant. They're not unusual findings. So multiplicity can show up with multiple subgroups. So we saw age and sex in a few slides ago, multiple endpoints or analyses. So subgroups might be effect on males, effect on females, effects on people over 65. Endpoints might be effect on blood pressure, effect on life expectancy, effect on happiness. And finally, if you even run the same different statistical analyses on the same subgroups and same endpoints, you may get different results. So for example, if your primary analysis is some sort of multiple regression, but use different covariates, you actually may get different results. So multiplicity can come up in a whole variety of ways. There are solutions to the multiplicity problem, and that the primary solution is pre-specification. You tell the world ahead of time what you will primarily look at. So in that study a few slides ago, if the study had primarily ahead of time decided that they're most interested in females of a certain age, and they found a statistically significant result in that subgroup, that would be an important finding. At least that would be a statistically significant finding. So this avoids what's known as fishing for p-values, looking at multiple subgroups or endpoints until you find something that has an interesting looking p-value. So the way you tell the world ahead of time is through protocols and statistical analysis plans. So those should be completed before the protocol should be completed before the start of the study, and the statistical analysis plan is usually completed before unblinding of any data. Now, there are statistical methods that can handle multiple outcomes or multiple subgroups. I mean, suppose you genuinely are interested in two different endpoints, like blood pressure and survival. There are statistical methods that can properly adjust just for these multiple comparisons and this multiplicity. But I certainly won't go into them today, but the important thing is they need to be pre-specified too. We spent a little time on variability, and now I'm going to move on to bias. So in bias, I'll cover various types of bias and say something about sensitivity analyses. This section will be longer than the variability section, but more intuitive, I hope. Okay, we'll go back to this formula for a z-statistic. There's nothing new here. We have a z-statistic that's the effect estimate over the standard error of the effect estimate. And for a given effect size, say 0.5 meters, if the sample size goes up, the standard error goes down, the z-statistics goes up, and the p-value goes down, meaning it's more significant. Let's look at a bias example. Here we have an unblinded trial, and the endpoint is effort dependent. I'm going to use the 6-minute walk example again, how far you can walk in 6 minutes. Because patients know what drug they're on, whether they're on the experimental drug or the control, patients on the experimental drug may try harder. This may not be a conscious bias. This may be more of an expectation bias. They think they're on some novel therapy, so they're going to be doing better. Let's say on the average that they do 0.5 meters better. Some may do more than 0.5 meters, some may do less, but on the average, this expectation bias makes people on the experimental drug do 0.5 meters longer in a 6-minute walk. Let's assume there's actually no drug effect here. Now, the important point here is this 0.5 meter bias will not likely be affected by sample size. Everybody will have, on the average, this 0.5 meter bias. It's going to appear no matter what sample size you have. So p-values will get smaller for larger sample sizes, even though the drug is not effective. We've learned something about addressing bias. Sample size does not address bias. A larger sample size will not get rid of bias in most cases. What you need is design and conduct. You use design and conduct to address bias. So some of the design methods are randomization, blinding, good outcome ascertainment. What I'd like to say is analysis cannot rescue poor design and conduct. Analysis can address some bias and explore sensitivities to bias, which I'll talk about at the end of the talk. So in order to help identify biases, it's useful to categorize them. So I'm going to categorize 3 types of biases, and it's not an exhaustive list, but it accounts for much of the bias you may see. The first is confounding. This has to do with baseline differences between treatment groups, and this is addressed by randomization. I'll go more into this. The second type of bias to have your eye out for is measurement bias, or the endpoint ascertainment. This is typically addressed by blinding and reliable and valid measures. I really won't be saying any more about this in today's talk. And finally, there's selection bias. This includes both baseline and post-baseline population issues, which I'll have a bunch to say about in a few slides. Confounding. So without randomization, there may be systematic differences, bias, when comparing people getting drug A and people getting drug B. This is known as confounding. Let's take an example of this. Suppose drug A may be given to older, sicker patients. Perhaps drug A has less side effects, and older and sicker patients can tolerate it better. So even if there was no difference between the effects of drug A and drug B, the comparisons may show that drug A has worse outcomes because drug A is made up of sicker patients. Again, this is known as confounding. Now, I already mentioned, randomization addresses confounding because it makes the 2 comparative groups comparable at baseline. Many of the studies you see in the paper are not based on randomized studies. So if you ever hear anything about coffee or red wine or chocolate, chances are it was not based on a randomized study, and I would take the results with a grain of salt. Now let's move on to selection bias, and I've broken that up into 2 types. So the first type is baseline study population is not representative of the intended population. This is a relatively easy concept to understand. So let's take an example. So suppose in your study there are a few people with comorbidities. So this will affect the generalizability or the external validity of your study, that what you study may not represent the larger population, in this example, people with comorbidities. The second type of selection bias is that the analysis population is based on post-baseline information. This is harder to understand, and I'm going to spend some time on this. So some quick examples that we'll dive deeper into are analyzing adherers only, analyzing those with no missing data, and analyzing those with an initial response to treatment. You can see that the population you're analyzing is all based on post-baseline information. What this does is it affects the causal relationships, the actual relationships you're trying to study, the drug effects. And this is actually an effect on internal validity. Even the relationships you estimate within your study are wrong, let alone that they don't generalize to a larger population. So here's a classic example of this from an old study, the Coronary Drug Project, that's often used to illustrate this point. So how adherence itself may be related to the outcome. So here the drug clofibrate is being compared to placebo for a mortality outcome. So on the bottom here, we have the percent of people, the percent of mortalities in the clofibrate group, which is about 18%, and the percent in the placebo group, which is about 19%. So there's not much difference here. It doesn't look like clofibrate has much effect on this population on mortality. But there's actually more to this table than I showed you initially. This table also breaks down the results by your adherence, whether you have less than 80% adherence or greater than 80% adherence. This is basically how much of the drug you took compared to what you were supposed to take. And what we see for the ones with higher adherence, those with greater than 80%, the mortality is down to 15%. And those with less than 80%, the mortality is close to 25%. So if you compare that 15% of the adherers, the people who actually took the drug a reasonable amount of time, to the placebo amount, it looks like the drug has an effect. But the thing to be revealed here is the placebo group has the same pattern. So the people who took placebo more than 80% have lower mortality than the people who took placebo less than 80%. Now, obviously, taking placebo more is not going to affect your mortality. What's really being demonstrated here is the people who have a higher adherence are different from the people who have lower adherence. They may have more attention to their own health, or there are any other number of differences between the two groups. And this difference, based on a post-baseline factor, can introduce bias, which doesn't show up if you do not divide by adherence, but when you start looking at adherence, you see that there are differences. So let's talk about adherence and missing data and biases. Patients who complete therapy may be different from those who stop therapy. I think we could think of several reasons for that. The people who complete therapy, again, may be more interested in their health. They may be less sick. They may have less side effects. So there's a difference. So treated group patients who complete therapy may be different from control patients who complete therapy. Now this is very subtle, and you might not be able to pick it up in the time I have on this slide, but the first bullet talks about if those who complete therapy are different from those who stop therapy. But this difference actually may be different between the two treatment groups. So if the first bullet was the only problem, then maybe you would just look at people who adhere to the drug. Like in the last example, those are greater than 80% adherence. But the reason people don't adhere may be different for patients on treatment versus those on placebo. For example, maybe this drug has a lot of adverse events associated with it. So the people prone to adverse events stop treatment. But on the placebo, those who stop it may be stopping it. They're not stopping it for adverse events because they're only taking placebo. They're stopping it for other reasons. So this is a rather subtle point, but it explains why we just can't look at adherers. Analyzing only patients who complete therapy, or what we call complete data, will likely produce biased results. So that's the kind of conclusion of all this. So let's talk about the different types of adherence and missing data, because this has implications on how you address them, in the design and analysis. So one type is patients may stop treatment. They may stop treatment, but you may actually be able to follow them up after they stop treatment. So stopping treatment is not the same thing as leaving the study. Leaving the study is the patient is not agreeing to have any more follow-up, or has just been lost to follow-up perhaps. So a patient can stop treatment and still be followed up. So I'd like to distinguish those two situations. And a third situation is, patients may miss a single study visit. That visit might have involved a particular treatment or a particular assessment, perhaps the endpoint assessment. So you may be missing some of your endpoints. So these are three different types, and it may be important to think of them separately and how you might deal with them. But they're all called intercurrent events, and they should be handled appropriately and not often the same way. So what causes, I've already talked a little bit about this, but what are some of the causes of lack of adherence or missing data? Well, the first thing might be random. So it's not related to anything about the patient's characteristics or the patient outcome. So if this was the only problem, then we could just ignore it, basically. The patients that have the information are just like the patients who don't have the information. It's all random. We could just use the patients where we have the information. We would lose sample size, but we wouldn't create any bias. Another possibility maybe is the frailty of the patient, like sicker patients drop out. And again, maybe it's lack of efficacy. Maybe The patients who are perceiving not getting any benefit from the drug are more likely to drop out. And also I've mentioned side effects. Maybe certain side effects makes patients drop out. Again, it's important to think about this as you design your study and as you come up with analyses and eventually sensitivity analyses. So just a word on adherence. I don't want to make it imply that adherence is forcing the patient to do something. This is not ethical. The protocol should allow for the best interest of the patient. So we're not trying to force adherence. We're trying, the study will represent what happens. We may encourage adherence, through education efforts or whatever, but we're not trying to force the patient to do anything that's not in their best interest or not that they're willing to do. So adherence is basically following the protocol, which should allow for the best interest of the patients, even if that means stopping therapy. Okay, so I've already talked about some reasons for missing data, some reasons for lack of adherence. So what are some of the solutions for that? The primary way we analyze efficacy trials typically is intent to treat, which is currently also known as treatment policy. So the first step is we want to get follow-up data regardless of treatment adherence. So even if someone stops treatment, we still like to get the follow-up data. So as I said earlier, stopping treatment is not the same as stopping the study or dropping out of the study. The patient may, once they stop treatment, may not choose to be in the study anymore, and that's their right and choice. But to the extent that patients are willing to continue to be follow-up, even if they're not getting treatment, has an advantage, because we can see what happens to these patients. So we analyze patients according to their random assignment, regardless of adherence of missing data. So this answers the question, what is the effect of being assigned therapy? So you may stop therapy, you may start another therapy, but it does answer the question, if you're randomized to this therapy, what is the effect? And importantly, it maintains the randomization, because you're still comparing people randomized to one group versus the other group. Now, there are problems with the treatment policy or the intent to treat approach. Patients may stop therapy, they may cross over to the other group therapy, or they may receive rescue therapy. So stopping therapy is pretty obvious, we already talked about it. In some studies, patients on the placebo group may actually start taking the investigative agent, and that could be because it's available through some other means or as part of the protocol. Or some protocol often will allow for rescue therapy. If a patient worsens, they don't allow it just to continue to worsen. Some sort of approved rescue therapy is used for that patient. So this creates sort of complications in interpreting this intent to treat, because these things may happen to a patient and may affect your view of like the efficacy of the drug, but are not accounted for in this ITT approach. The ITT approach is basically what it means to, what the effect is to be assigned therapy, not what actually happens to you. And unfortunately, you still may have missing data. So ideally, if a patient stops therapy, we would recommend that you continue to collect data as they would if they were still on therapy, or maybe some abbreviated version, or whatever it takes to get the endpoint you need. But a patient may choose not to continue to be followed up, or they may be lost to follow-up. So you still may have missing data. So if to do the intent to treat, you need data on all the individuals. So this is sort of a limitation. So you'll have to find some way of handling this missing data. One approach, not so much for the missing data, but for some of the issues in rescue therapy and stopping, is use a composite approach. So a composite approach would treat stopping therapy, changing therapy, or the use of rescue therapy as a failure in a composite endpoint. So suppose your endpoint was some sort of clinical response. a success or a failure. So the composite would count as a failure not only whatever is being measured, but any of these events like stopping, changing, or rescue would be considered a failure as well. So that's one way to kind of address some of these issues of, that placebo patients may have got rescue therapy, so they're now going to look better. But if you consider that a failure in this composite approach, the ITT approach still works and it still maintains randomization. Again, it doesn't address the missing data, but I'll get to that in a bit. But in general, the approach should be tailored to the specific clinical question you have. Now I'm going to say something about missing data, but mainly uses as an example of sensitivity analysis. Sensitivity analyses involve varying assumptions to explore the potential biases. And the setting we're going to use is the drug effect for patients with missing data is not the same as the drug effect for patients with complete data. I've already said that we don't see bias in the data, but we can use sensitivity analyses to explore assumptions on the bias. This is a rather complicated figure, so I'll spend some time on it. It comes from a study where the primary result was statistically significant, but like all studies, there were missing data. So the green areas in this plot are under certain assumptions where the drug is still statistically significant, and the red areas are where the drug is no longer statistically significant. And there potentially could have been yellow areas where the placebo was actually statistically significant, but that didn't occur. So what we're seeing here is various assumptions of the missing data. The x-axis is assumptions on the estimated mean of the endpoint for those with missing data for the treated group. And likewise, the y-axis, the vertical axis, is assumptions for the estimated mean of the control group with missing data. So most of the patients are not missing data, so that they're still in this analysis. But for the patients that are missing data, we make assumptions of what their mean value would be. And we see different regions where the study is still statistically significant. So this involves clinical interpretation, like what would be reasonable to assume for these missing data. If you can make somewhat extreme assumptions and still see a statistically significant result, then maybe the bias from the missing data is not a big deal. So this involves, again, clinical judgment, but allows you to explore different assumptions for the missing data and maybe see how strong the primary result is. This brings me to concluding statistical principles. In summary, careful design and conduct are needed for reliable results. Randomization addresses baseline biases. Blinding, good adherence, good follow-up, and good endpoint ascertainment addresses post-baseline biases. Pre-specification is important to address multiplicity. Here are my take-home messages, returning to the theme of the beginning of the talk. Address the clinical question, minimize bias and minimize variability, and careful design conduct will promote valid and reliable results. Challenge question number 1. Which of the following does not reduce bias? A larger sample size, randomization, blinding the knowledge of the drug from the study participants and investigators, pre-specification in the protocol and statistical analysis plan. The answer is a larger sample size. If we go back to that example in the unblinded trial with the effort-dependent endpoint of the 6-minute walk and the belief that patients on the experimental treatment may try a little bit harder, on the average, every patient is going to have that 0.5 meters extra effect on the experimental arm, and that bias is not going to go away with larger sample size. Randomization clearly addresses confounding, and blinding clearly addresses endpoint ascertainment, so they do address bias. And I'll leave pre-specification for a moment. The second challenge question is, which of the following addresses multiplicity? A larger sample size, randomization, blinding the knowledge of the drug from the study participants and investigators, pre-specification in the protocol and statistical analysis plan. Here the answer is pre-specification in the protocol and statistical analysis plan. We talked about multiplicity, that if you look at enough things, you're going to see something statistically significant. So pre-specifying what you're primarily interested in will avoid that issue. So with say thank you, and I'm looking forward to answering questions in the question and answer period.
Thanks so much to both Jim and Mark for getting the course started. We've covered a lot of territory in a short amount of time. This is terrific. So we do have some questions that have come through. I'm going to go ahead and start with Jim. The first question, you mentioned that the study population may be narrowly or more broadly defined. Other than enrichment, can you say more about considerations for each of these?
Sure, Kim. Thanks a lot for passing along that question. Yeah, so the study population can be narrowly defined at times or more broadly defined at others. And I think I gave some examples of this, but I'm happy to embellish on it a little bit. So I'm going to think about it from the context of a typical drug development program here, but the principles could apply to other clinical research. So early in drug development, when a drug is first being studied, for example, for a particular use, one is probably doing trials that involve a fairly small number of patients to test a drug, perhaps to explore certain endpoints, to get proof of concept, figure out whether this drug candidate is worth developing further for a particular use. And so really one is trying to identify a signal and really find out whether or not, there's a signal here to carry that drug further in development. And so when we think about how to best define a signal, you can either have a drug with a very large effect, or what's more common is to have a drug that might have a smaller effect, in which case you really want to be able to distinguish it from the noise. And so reducing noise or reducing variability becomes particularly important to try to help identify a signal. And so in early development, one approach to reducing variability is to more narrowly define a patient population such that there is more homogeneity in the population that you're exploring the drug in. And there's obviously limitations to that, and that is you're answering questions about a fairly narrow population that, as the drug is then carried further in development, you might realize that it does not work as well as you hoped once you broaden the population out. But that's one example. Another example could be from a safety perspective. Early in development, when there's not as much known about a particular new drug, you might have very real and legitimate concerns about exploring that drug in patients with certain comorbidities or other complicating features, because you're trying to get not only to protect the patients before you have more safety data accrued, but also it might be more difficult to discern what that drug's safety profile is, on a background of other comorbidities, other, concomitant medications and whatnot. But as I mentioned, as a drug goes further along in development, one is not going to want to continue to restrict the population to these fairly narrow groups, but rather really broaden out the population, so that by the time one is putting together an evidence package from both an effectiveness and a safety perspective, the drug has really been studied in the patients for whom you would expect to take the drug if that drug were to be approved. And I would think that the same principles would apply elsewhere in clinical research, that you really want the results from the clinical investigation to be generalizable to whatever the clinical research question that you've asked, and not only applicable to, for example, a very narrow subset of individuals. So hopefully that helps answer the question. Thank you.
Thanks, Jim. And I have one follow-up question that is related. Would the use of enrichment in selecting the study population lead to a, quote, tighter label?
Thanks, Kim. That's a good question too, and you're right, very, very related. So if one were to progress in drug development and use various enrichment strategies, and you generated evidence from clinical investigations in a particularly enriched population, the question is really what scientific conclusions can then be drawn from that, which is really how, if you're seeking drug approval, how labeling would be written, because the labeling should really reflect the use of the drug as it's been demonstrated to be safe and effective. So one example would be, and there's really, I should say, there's no just rules of the road here. Sometimes, and by that I mean that it's very case specific. So, but I'll just give a couple examples that will hopefully illustrate the concept. So let's say one is using a predictive enrichment strategy where you have a drug, as I included in my challenge question, that perhaps has a mechanism that is targeted to a particular, gene variant of a disease. Well, one would then expect for that, the presence or absence of a particular variant to be predictive of whether that drug would work. And if that is in fact the case, then the labeling would very appropriately be limited to the patients for whom you expect the drug to work. And this can be a little bit difficult because it's not always obvious. And sometimes perhaps data are gathered on a biomarker positive, for example, population, and based on very good and solid hypotheses, and you don't have as much data in the biomarker negative population, and perhaps the drug is actually still effective, just less so in biomarker negative patients. And so really the extent to which one actually has data on those populations to see if there could be an interaction between, for example, biomarker positivity and the treatment effect, would all play into decisions about how the label might be written. With respect to prognostic enrichment, it's also not, there's no one size fits all answer. Sometimes it's very reasonable to enrich a trial for a high risk population because events will happen faster, but there's no scientific reason perhaps to think that the drug would not be effective in patients who are a bit lower risk, maybe moderate risk or whatnot. That maybe the effect size or the absolute risk reduction, for example, would be smaller in the low risk patient. And those are really scientific considerations that one would have to think through. I can think of examples, where prognostic enrichment has led to a labeled indication that is exactly the population that was studied in a trial, and other situations where it was believed that the indication could be broader and not limited to the quote unquote high risk patients, but other sections of the labeling would describe the enrichment strategy, so that providers would have that information should they choose to take a look at it. So hopefully that helps, and thanks again.
Great, thanks, Jim. And Mark, we have a couple of questions for you here as well. First, can you say something about what makes a good endpoint in a clinical trial?
Thank you for that question, Kim. Yeah, no, that's a very good question. Dr. Jim Smith addressed some of that in his talk, that first and foremost, it should address the clinical question. The endpoint should be chosen to get at what question you're asking for. But there are definitely more considerations that come into play. It should be what we would call sensitive to the drug. So, for example, or sensitive to the perceived effect of the drug. For example, if you're maybe studying an antidepressant and you don't expect to see an effect by week 2, but maybe the mechanism of action or past experience would lead you to believe, excuse me, you would see effect maybe at week 12, then obviously week 12 is the more appropriate time point for the endpoint. So you're trying to look for something that would be sensitive to the hypothesized drug effect. Now, it still has to be clinically relevant. If reversing that example, if the effect only had an effect on if the drug only had an effect on depression at 1 week and no effect beyond 1 week, well, you might be able to show an effect, but it probably wouldn't be very clinically relevant. So again, the first criteria has to be clinically relevant. But moving on from that, being sensitive to the hypothesized drug effect is important, and then the bias and variability come into play. As I said, you're always trying to minimize bias and minimize variability. Here, variability would mean, like, if for some reason you were theoretically able to take the measurement twice, would you get nearly the same result, or is it going to be very variable? Some endpoints are just naturally going to be not very variable. Maybe a heart attack is a very hard endpoint, and how you measure it, if you were to go back and measure it, take the labs and EKGs, you'd get the same answer. Other endpoints may be more variable. Endpoints based on maybe a patient-reported endpoint might be more variable. If the patient took the same answered the same questions again, they might come out a little differently. But again, you have to foremost stick with the clinical relevance. So variability is important, but you don't want to answer a question that's no longer relevant. Just one kind of closing thoughts on this is FDA does have a series of guidances and draft guidances on patient-focused drug development. They're mainly focused on the issues of these, maybe something like a patient-reported outcome, although there's other examples in those as well. But they do go into a lot of what makes a good endpoint, both from a clinical relevance point of view and including bias and variability. So I hope that kind of gets at some of at least my thoughts on what makes a good clinical endpoint.
Great, thanks, Mark. And one additional question for you. If a study is not statistically significant, what can we conclude? Can we say that the drug has no effect?
Okay, another good question. I mean, this is a question that goes beyond even clinical trials, but I think it's an important thing to consider. So the concept of statistical significance is a tricky concept, and that's unfortunate. That's unfortunate for you non-statisticians, and it's unfortunate for a statistician. It means, as I tried to explain in the talk, that you've observed something that's very unlikely if the drug had no effect. So therefore, you conclude it probably did have an effect. So when you have a statistically significant effect, you can generally conclude it has the drug has an effect. I mean, of course, you have to consider all other issues of biases that may have resulted in this. But let's assume it was a very clean, well-conducted, simple trial, and you see a statistically significant effect. Great, you see effects, so you say the drug has an effect. Now, the converse is more tricky. If something is not statistically significant, you cannot automatically conclude that the drug doesn't have an effect. There are several things that may come into play here. Maybe the most important to consider is what I tried to get at is statistical power. It may be that the study had very little power to detect an effect. So even if there was an effect there, the power to detect it might have been very low. The power is a number, is a probability itself. So it can go from 0 to 100%, although it probably can't be either 0 or 100. But typically we design clinical trials that have 80 or 90% power for a given effect. So if we think the effect, say, is 2, whatever that might mean, we have 80% or 90% chance of detecting that. But if we run small trials or trials that are underpowered, we may not detect real effects. So the lack of statistical significance doesn't necessarily imply that there's no effect. Now, if the trial had a lot of power, that's already giving you evidence that if you didn't see it, then it probably doesn't have an effect. But maybe a more straightforward way to examine that is to look at confidence intervals, which I didn't go into in my presentation. But a confidence interval gives you the range of likely effects. And at the upper end of the likely effects is still a very small effect, then you might conclude that the drug doesn't have much of an effect. So again, I hope that addresses this question. Thank you.
Yes, thanks, Mark. And I think we may have enough time for maybe one more question for each of you. Let's go back to Jim. Is placebo the same as no treatment? If not, what's the difference?
Thank you for giving me a chance to clarify this. So absolutely true that placebo and no treatment both mean that someone is not getting an active drug. The difference between a placebo-controlled trial and a no treatment controlled trial is really blinding. So in a placebo-controlled trial, as I described, and I think as folks are probably well familiar, that is going to be a blinded trial because otherwise you're giving participants a placebo and telling them, hey, I'm giving you a placebo. That might not be the effect that you're intending to have and could introduce bias into the trial. So a no treatment control trial is really when blinding is not possible and it's going to be an open label trial anyway. Then you still have a control arm that's just getting standard of care, and then your interventional arm is likely getting standard of care plus the open label intervention. So hopefully that clarifies. Thanks.
Great, thanks, Jim. And Mark, one more question for you. How can data fudging, that's a quote, be avoided, and how can you detect if it happens?
Okay, another good question. And I think during my talk, I probably gave the primary way of avoiding it, but I'll go deeper into this. That to avoid data fudging is really a problem of multiplicity. I assume that's what's meant here. I assume it's not meant like making up numbers or so. I think what's meant here is that you've looked at a lot of results. Maybe you've conducted a lot of studies, and just like my example, you found one subgroup perhaps that was statistically significant, and then you write a paper on that, and you don't mention all the other things you looked at, and you just mention I have a statistically significant result. That's what I think is meant by fudging here. And as I said in the talk, the best way to avoid that is pre-specification. That you have a protocol in place, and hopefully in a statistical analysis plan if the analysis is non-trivial. And this way, if you set out ahead of time what you're most interested in, and if you find that, it's an important result, and if you don't, that might be interesting to report as well. But if you start reporting on other things that you didn't set out to look at, you have to come clean about that this is an I think Dr. Jim Smith went into this. This is an exploratory endpoint. Although the primary endpoints were not statistically significant, or maybe they were, this exploratory result might be hypothesis generating that we might want to explore in further studies. Now in practice, this is a group for clinical trials. I think it's relatively straightforward. I think there may be requirements for publicly registering your protocol ahead of time, so the public knows what you're looking at. I'm not personally familiar with those requirements, but that's one way to be clear what you're looking at. I mean, you could run a lot of studies and not tell people you're running a lot of studies, and then just pick out the one that's significant. That would also be kind of an issue with fudging, even if you had protocols. I think it's an unlikely scenario for clinical trials because of they're expensive and time-consuming to run. You're not running a lot of trials and just picking out the one that looks best. But I did point out for observational studies, you might see a lot more of that. Now, this group might not be generally focused on that, but it's something to be aware of from kind of a scientific point of view. So thank you. Thanks for the question.
Thank you, Mark, and thank you to Jim also, both for your presentations and for your willingness to engage with the audience with questions. And thank you to the audience for your attention and for submitting some questions for us. We are going to actually go on our first break. And we will resume presentations at 1:45 p.m. Eastern Time. Thank you all.